Good evening, everyone. Today I will be presenting my senior research project on the effect of natural ingredient-based mouthwash on Streptococcus salivarius. Before we begin, I'd like to introduce myself. My name is Marenah Smith. I go to George Washington High School. In the fall, I will be attending Virginia Commonwealth University to major in biology to pursue a career in pharmacy. Because of my interest in health and medicine, I chose this topic. Oral health is affected by our oral hygiene. Each day we use products such as toothpaste and mouthwashes that contain ingredients that can be harmful to the oral microbiome. Personally, I'm not a fan of commercial mouthwashes because of the burning sensation and the taste. This brought me to ask the question, is there a mouthwash I could create myself that satisfies taste, convenience, and effectiveness? This topic applies to those with oral sensitivities and diseases who are unable to use alcohol-based rinses because of side effects like burning, taste disturbance, and irritation to open wounds. Mouthwashes containing natural ingredients are safer, healthier, and more convenient than alcohol-based rinses. In order to carry out my experiment, I had to first review literature pertaining to my topic. Mouthwashes come in different chemical compositions. The most common we see are Listerine, Colgate, and Crest. These mouthwashes contain alcohol and menthol, which contribute to the burning sensation. Chlorhexidine is another common ingredient found in these mouthwashes that wipe out beneficial microbes in the oral cavity. Commercial herbal mouthwashes contain lots of essential oils and filler ingredients. A downside to these rinses is the increased amount of sugar being coated on the surface of our enamel, which contributes to plaque. Homemade mouthwashes are formulated with solely natural ingredients so that people know exactly what they're ingesting. Consumers are also able to control exactly how much of something they include in their mouthwash. This is imperative to individuals with oral sensitivities and allergies to specific ingredients. Out of all the literature I reviewed, the most frequently used ingredients were peppermint oil, basil, and red ginseng. Peppermint contains antibacterial properties that are used to freshen breath and prevent the development of cavities. Basil, shown in the top image, is found to reduce plaque and gingivitis because it works as an antioxidant, which reduces the risk of tooth decay. Red ginseng, shown in the bottom image, is a plant native to Asia. Ginseng extracts inhibit inflammation and reduce oxidative damage to cells. As we know, the purpose of mouthwash is to kill oral bacteria. The most common strain of oral bacteria is called Streptococcus mutans, shown in the top image. In the literature I reviewed, researchers primarily tested this specific strain. Streptococcus mutans is known for binding to the tooth enamel and progressing into dental caries. Dental caries are formed when this bacteria metabolizes sugars to produce acids that break down the enamel, causing tooth decay. My study aims to bridge the gap between the testing of oral bacteria strains. The bacteria used in my experiment was Streptococcus salivarius, shown in the bottom image. It is found in the oral cavity where it binds to the epithelial cells on our tongue and inner cheek. This bacteria is responsible for halitosis, known as bad breath. After reviewing the literature, it brought me to ask the question, how effective are natural ingredient-based mouthwashes in reducing Streptococcus salivarius? After developing my research question, I developed my hypothesis. My research hypothesis states, if the mouthwash contains herbal ingredients, then it will exert some form of efficacy against Streptococcus salivarius. My null hypothesis states, if the mouthwash contains natural ingredients, then it will have no difference in efficacy against Streptococcus salivarius. Moving on to my variables, my independent variable was the different herbal mouthwash formulations. The dependent were the zones of inhibition, the constants were the type of bacteria, the incubation time, and how the diffusion discs were saturated. My control was the bacteria group only being tested with distilled water. To begin my procedure, I gathered the following ingredients: distilled water, vinegar, lemon juice, pomegranate juice, peppermint extract, mint, basil, rosemary, baking soda, green tea, and cinnamon. The materials I used were measuring spoons, pipettes, swabs, forceps, and a ruler. The first step was to assign each ingredient a number and randomly assign each mouthwash 3 ingredients. My sample size of 30 is comprised of 3 experimental groups, 1 control group, and a comparison group of Listerine. In total, I had 5 groups with 6 petri dishes each. Next, I combined the ingredients. Each mouthwash comprised 2 tablespoons of water, 1.5 tablespoons of the liquids, 1/4 teaspoon of peppermint extract, 1/2 teaspoon of the dry ingredients. Group 1 consisted of vinegar, mint, and rosemary. Group 2 was lemon juice, basil, and green tea. And Group 3 was pomegranate juice, baking soda, and cinnamon. After their solutions were made, petri dishes were labeled based on their group. Sterile swabs were used to transfer bacteria onto the dishes, rotating the dish 90 degrees to gain maximum coverage. The diffusion discs were then saturated with each solution and placed into each quadrant. The petri dishes were sealed and incubated for 48 hours at 37 degrees Celsius and refrigerated for an additional 48 hours. After incubation, I measured the zones of inhibition around each disc using a ruler. I compiled my raw data into an Excel spreadsheet and conducted my statistical analysis. For my statistical analysis, I used the one-way ANOVA test in order to compare the means among my mouthwash groups. At the significance level of 0.05, the null hypothesis was rejected with a p-value of 0.002. The images you see on the slide depict bacterial growth and the zones of inhibition. Going left to right, we have groups 1, 2, and 3. To give you a brief refresher, group 1 consisted of vinegar, mint, and rosemary. Group 2 was lemon juice, basil, and green tea. And group 3 was pomegranate juice, cinnamon, and baking soda. The bar graph shown represents the direct comparison between the mean zones of inhibition measured from each mouthwash group. We can see that mouthwash groups 1 through 3 and the comparison group of Listerine exhibited significant efficacy in the susceptibility of the bacteria. Groups 2 and 3 displayed the highest zones, while the control displayed the lowest. Based on this depiction of data, we can conclude that natural mouthwashes are effective in the bacteria resistance against Streptococcus salivarius. In conclusion, at the 0.05 level of significance, there is sufficient evidence to conclude that natural mouthwashes are effective in reducing Streptococcus salivarius. The results from my study suggest that natural ingredients are effective in oral bacteria reduction. To consumers, this shows how useful herbal products can be in treating oral deficiencies. Consumers are exposed to a broad selection of ingredients they can use to formulate their own mouthwashes. Consumers are able to focus on their personal needs in a mouthwash and narrow down ingredients to target these needs. Herbal ingredients create a vast opportunity for mouthwash users to customize their own solutions based on taste and effectiveness. Although my experiment was successful, there were 2 factors that could be altered to give the experiment different results. The first was the type of bacteria and the collection of it. Since the oral cavity is a microbiome composed of many different bacteria strains, swabbing the mouths of participants may yield more significant results than using Streptococcus salivarius alone. The second factor were the ingredients I chose to use. I used cheap and accessible items, which limited the amount of variety in the mouthwash formulations. If I used ingredients that were more exotic and expensive, the data may have been more varied. The results obtained from my study can be further researched through the field of periodontology. Periodontology is the study of teeth structures and diseases or conditions that affect them. Researchers can use these findings to expand their knowledge of herbal ingredients and how they react to underlying oral conditions. The ingredients used in my study can be used by researchers to formulate new herbal mouthwashes. In turn, these rinses are capable of treating oral diseases such as plaque and gingivitis. As this experiment was performed on Streptococcus salivarius, it can be replicated to test other bacteria strains. The more we know about the reduction among oral bacteria in natural products, we are able to conclude what is most beneficial to the oral cavity. During this research process, I learned several things. The first being the unpredictability of bacteria and its growth. Originally, incubation time was only set for 24 hours. However, after one day, there was absolutely no growth. After an additional 24 hours, some dishes were covered in large cultures and individual colonies. The next thing I learned was how many common ingredients held significant antibacterial properties. All of the ingredients I used were easily accessible and often already found in your kitchen. The entire research process taught me the importance of accountability. I was responsible for taking the proper precautions to ensure my experiment was performed correctly. This included adopting problem-solving and organizational skills that will be used in my future endeavors during college. Before I end my presentation, I'd like to give a big thank you to Ms. Long for walking me through the entire research process, and to Ms. Dillon for aiding in the methodology portion of my study. I will now be taking any questions. Ms. Probst.
In your literature review, did you find any information on, there's some people that are against mouthwashes because they rid your mouth of normal fluorides such as nitrous oxide that we need for certain processes in our body? So in the literature review, did you see that? And if you did it lean more towards natural mouthwashes? And did you use a recipe for these? So I guess I have 3 questions.
Okay. So the first question Ms. Probst asked were, did I find any studies talking about people who are against using mouthwashes? And the answer to that one is no. All my studies focused on using mouthwashes. Okay, so what were your other 2 questions?
So that takes care of the first thing.
Okay.
Okay, the third one is, was the recipe that you used for the natural products, or did you find that in your research or separately?
Okay, so the next question she asked were, did I follow a recipe? So I did not follow a recipe. I gathered all the ingredients based on what I had at home and then researched the additional products such as the green tea and the lemon juice, and I just randomly assigned them a number and made my own formulations.
Okay, I was just wondering like how, people would like to use vinegar as a mouthwash. I mean, was it pretty diluted with the other stuff, or did you remember?
It wasn't diluted at all. It was like 100% vinegar.
Oh, okay.
Mr. Shaw.
Looks like you used dry basil and rosemary as opposed to fresh. What was like the rationale behind that decision?
So Mr. Shaw asked why I used dry basil instead of fresh basil and herbs. So I use this because of convenience and because I already had it, but if I were to replicate the study, I would use the fresh herbs to get more accurate results.
How do you think it would have changed your results if you were to use the basil if that came up in your research?
So he asked how would using the fresh herbs make a difference in my study. I think the fresh herbs, they have more of the properties instead of the dry basil. It kind of rids it of all its properties, so using it fresh would give it the full potential it has to work.
So I noticed that you had different levels of effectiveness for the 3 different varieties of combinations that you used. I don't remember which one was the most effective. Can you go back to the mouthwash 2? What was the combination for that?
That was lemon juice, basil, and green tea.
And do you have any idea of why that was more effective than the others?
Yes, so Ms. Waltz asked why group 2 showed the most efficacy compared to the other groups. I would say because this one contained lemon juice, which contains a lot of acidity, it rid the bacteria the most. Mr. Hare.
You did not have participants actually try the multiple types, correct?
Yes, so Mr. Hare asked, did I have participants? And no, I didn't have participants. I used the bacteria directly on the dishes.
So next time we would have all loved to try it, so just.